With no further ado, I'd love to introduce Dr. Brendan Curti, our first presenter. Dr. Curti started his career in tumor and T-cell immunology back east at the NCI's Biological Response Modifier Program in 1990. Back when I was still in high school. He joined the Early Childhood Research Institute at Providence Cancer in 2002. He is the Robert W. Franz Endowed Chair for Clinical Research and Director of the Melanoma Program at our institute. He works every day with researchers to translate basic or bench science into clinical trials and improve care for patients with melanoma and other advanced malignancies. He treated his first patient with adoptive cellular therapy back in 1990. Since then, he has authored and managed dozens of first-in-human clinical trials encompassing cellular therapy, cytokine therapy, immune co-stimulators, and cancer-attacking viruses. His talk today is on tumor-infiltrating lymphocyte therapy in melanoma. Dr. Curti.
Thank you, Chris. Again, thank you for the introduction. Getting over a little bit of a cold. I promise no one can catch my cold from participating in this seminar here today. And as you've heard, I'm going to talk about ACT, or adoptive cellular therapy. As is true with a lot of technology, there's a lot of jargon. I will do my best to define what those terms are as we proceed with our discussion here today. And I also take every opportunity I can to include puns in titles for my talks. And so TIL, or tumor-infiltrating lymphocyte therapy, the future, ? And we'll see as we have our discussion here today. So these are my disclosures, not relevant to the topics today. And so here are some questions that I'll pose and do my best to answer this afternoon. And so really, what is adoptive therapy? We bandy this term about, but what really does it mean? What are the main components of this type of treatment? What sort of immune cells are best to kill cancer cells? And I'll hint at the final answer, which is we don't really know, although we have some ideas as to what the best candidates are. My follow-up question is, how do these cells actually kill cancer? That's another question to which we don't have a full answer, but I'll just say most of us think that it's CD8 T cells, and I'll talk more about those later. But I'll say there's room for other cells and other immune therapies as well. So putting all of this together, of course, we'll talk about some early efforts and how our current procedures and clinical treatments have come together and what all of the moving parts are. I'll talk about some recent results that led to the FDA approval of lifileucel for certain patients with advanced melanoma. And I'll specifically talk about the individuals who for whom this therapy could be offered. So this idea of adoptive therapy goes back well into the mid-century part of the last century. And so this term was first used in the mid-1950s. And this comes from mouse medicine and an observation that if you took immune cells from lymph nodes in mice that had rejected a tumor and took those immune cells and put them into another mouse, the mouse that received the cells could reject the same tumor, but not a different tumor. And so in some sense, the new mouse had adopted the cells from the mouse that already had immunity. And although there was a knowledge of immune cells and T cells and B cells, that knowledge, as you might imagine, was not all that sophisticated in the mid-1950s, but it was a robust concept that has lived to the present day. And so again, immunity in a transplant model could be transferred from a donor mouse and adopted by the recipient mouse. And so when did the first human get adoptive cellular therapy? Well, if you count allo transplant, so basically bone marrow transplant, and receiving bone marrow cells from an individual who has the same tissue type, then the answer is 1957. So again, a technology that goes back over 65 years. The first solid tumor adoptive cell therapy clinical trials were done in the 1980s, starting with so-called LAC cells. LAC stands for lymphocyte-activated killer cells. The first TIL patient, or tumor-infiltrating lymphocyte patient, was treated in the 1980s and is still alive, by the way. The FDA granted accelerated approval in 2024 for this technology. So after 40 years of work, this has finally come to fruition. And we'll talk a lot more about that coming up. So there are a great many moving parts to this therapy, and it starts out with a surgeon removing a melanoma deposit from the patient's body. The melanoma that's removed by the surgeon has to be of a certain size because you have to get a certain number of immune cells at the end of the processing. After the tumor is removed from the patient's body, it's taken to a lab, and the tumor is actually chopped up into little fragments, and it's grown in certain conditions with certain immune substances, such that the immune cells grow, but the melanoma cells die. And after that is done, you usually get a number of immune cells, T cells specifically, that are in the billions. So you start out with a few thousand or perhaps 100,000, and then you get billions after the processing. The patient also has to be prepared to receive their own cells back, and this involves lymphodepletion or chemotherapy, basically to lower blood counts. The chemotherapy medicines, we'll talk more about this later, but the specific ones are cyclophosphamide and fludarabine. After that, the patient gets their own immune cells transferred back or adoptively transferred. They then get interleukin-2, which is the strongest known growth factor for T cells. And this helps their recently adopted cells to make copies of themselves to proliferate and also to make them cytotoxic. In other words, have better ability to kill the melanoma cells that may be remaining in the patient's body. There are a lot of side effects, though, that need management. Some of them relate to the chemotherapy itself. Some of it relates to the tumor breaking down. That can be a good thing, but also a complex thing medically. There can be a number of immune events that also come into play, and those need to be understood and managed. This is a cartoon that depicts all of the words that I just uttered. And so for those who are more visually oriented, but you can see in the top left, a melanoma, a tumor deposit can be removed. It can be on the skin, but it could be in a lymph node or lung or liver. There are other complexities, as you might imagine, from doing more invading or invasive surgery to do a tumor harvest. The tumor specimen taken to the lab, chopped up, grown in this case with interleukin-2 and something called OKT3 or anti-CD3. I won't go into that minutia, but just to say there are different substances that the T cells need to become active and to proliferate in the lab. The patient gets their cells back. This slide also depicts some extra steps, which are more experimental. And so I won't go into that, but just to say that there's a lot of effort to pick the winner or the cells that are very most likely to have the abilities to kill the melanoma cell. The patient gets their cells back, and as I mentioned before, they also get some interleukin-2 after the transfer of the cells. So I mentioned a couple of times this idea of conditioning regimen or lymphodepleting chemotherapy. So what the heck is that, and why do we do it? Because the chemotherapy itself really does not confer any melanoma fighting benefit, at least not best we know, but it can kind of reboot or reset the immune system. And so there are a number of different mechanisms to that. You can reduce inhibiting, also known as regulatory or Treg T cells. Pardon me. Chemotherapy will increase physical space in the bone marrow. As the bone marrow recovers from the chemotherapy, it makes a number of substances that are basically like vitamins for the T cells. They can promote the growth and the persistence of those T cells in the body. And again, the chemotherapy really doesn't kill melanoma. That's not part of the rationale. There's a lot of, I'll say, tradition to these regimens, and some early observations, actually some made back in the 70s and early 80s, show that low doses of cyclophosphamide, a medicine that's been used since the 1950s to treat cancer, but low doses of that medicine can, again, have this effect on regulatory T cells. The dose that we use now most often in lifileucel, well, most often in clinical trials, but also the standard with lifileucel adoptive cell therapy was actually used in the 1980s and is unchanged. And this was the same regimen that I learned as an oncology fellow in the late 80s and going into 1990 when I started my career at the NCI. So that part has been held constant throughout the history of this treatment. Pardon my cough. Let me talk a little bit more about those early results from the 1980s, in this case published in 1988. I remember presenting this article as an oncology fellow because of how eye-opening it was in that era. And you have to understand this in the era where there were really no proven therapies for patients with melanoma. So in this study, there were 20 patients. They had, as is typical for melanoma patients, disease in basically all organs of the body. As you can see, they had chemotherapy, immunotherapy. Even in this era, they got I'll say billions of cells, and understanding the lab processing was just being figured out kind of on the fly. But it was a pretty good first efforts, because these are still the same sort of cell numbers that we get today from the cell processing. The composition of the cells has changed, more CD8s compared to CD4s. I realize this is a lot of insider baseball, but just to say, in immunology circles, CD8 T cells seem to confer the strongest anti-tumor effects, but we shouldn't forget about some of the other immune cells either. And so this is a depiction of response. And I also offer this a little bit for the quaintness. This is a quaintness of this tumor response. Nowadays, of course, there are PET scans and circulating tumor DNA and all manner of other sophisticated means of assessing patient response. But here's a chest X-ray showing large melanoma deposits in the lungs that disappeared in this patient, and as you note, in under 6 weeks. And so, again, this was a very amazing finding given that era and the absence of other treatments that could do this. So overall, about half the patients responded. They responded at different organ sites. The duration of response was not that long, depending on your point of view, but the best responder, as I mentioned before, still alive and well, 36 plus years after the treatment. So these are the final words from that paper. I won't read them all to you, but other than to say it was a lot of work, and the authors concluded that this work should continue. And my response to that is indeed. And it only took, again, 40 years to make this a workable therapy. I'll pivot now to modern times. And so this is work from the last, I'll say 7 or 8 years. And these are the two studies that led to the approval by the FDA for adoptive cell therapy in melanoma. The two studies are actually the same study. And so in the upper left are the initial patients, and the lower right is the publication of all of the patients, including those that were described in the initial study, but those who were added on during the conduct of the study. I show this in part to show how clinical trials evolve in a way, and again, that'll be a little bit more clear as we head forward. And so these were all patients who had advanced melanoma and who had received modern therapy, so using medicines like nivolumab or pembrolizumab or ipilimumab, and also the various oral targeted therapies if their melanoma had a so-called BRAF mutation. There were 189 patients enrolled. 153 of those individuals received T-cell therapy. Some, however, even though they signed up, did not get the cells. Some of that was due, unfortunately, to the very rapid growth of the melanoma. Some were due to TIL production failures. So despite meticulous lab technique, it was not possible to grow the immune cells outside of the body. I'm casting no shade whatsoever at those who were doing this work. It is very difficult work to carry out. And 3 individuals opted for other treatment options. Again, they received the traditional lymphodepletion with cyclophosphamide and fludarabine. They also got a medicine known as Mesna or Mesna, depending on where you learned your oncology. But the Mesna is something that protects the bladder from the toxic effects of the cyclophosphamide. And so, unfortunately, the cyclophosphamide at high doses can cause injury to the bladder, and this medicine helps to prevent that. After the cells were infused, the patients got interleukin-2 or IL-2. IL-2 is an immune substance that all of us make, all of us have in our bodies, but when we give it as a medication, it's given in amounts many, many thousands of times greater than the body can make on its own. And in so doing, again, it can help these T cells to live long and prosper in the body. The way that the IL-2 is dosed is a little bit different than traditional interleukin-2, and so a few of you on the call may know me as the IL-2 guy, and so that's not my, I'm not here to go into that long and complex history, but just to say that the IL-2 with cell therapy is a little bit kinder and gentler in large measure because fewer doses are given. And it's also safer to give, especially in places that aren't highly familiar with this complex form of therapy. So I'm going to just show you some of the results, and this is a so-called waterfall plot. So any bar that is heading down is actually good. So that is indicative of a shrinkage of tumors. Bars that head up imply a growth of the tumors and their measurements. And so this is the early effort, and this next slide is the entire patient population. And just toggling back and forth, you can say these graphs have about the same shape. And so what I would conclude from that is that the results held up as the study got bigger. That's not always the case, but that was the case here, and that's a good thing. Of course, in the study, they also looked at survival. And so this is the early part of the study. And unfortunately, as these curves go down, that indicates, unfortunately, that patients have succumbed to their melanoma. But in light of the fact that all of these patients had progression of the disease after our current state-of-the-art immunotherapy, seeing patients survive 6 months, 12 months, even longer in some individuals is a good thing. And again, this is the survival from the larger study. It's a little bit different compared to the initial one. And so again, that just points out the nature of clinical research. The more patients you treat, you get a stronger and more robust signal. But again, the overall shapes of the curve are the same, and clearly patients are surviving. Not everybody, but a significant number are surviving longer as a consequence of receiving the cellular therapy. So this is just showing the same thing in a table. Roughly a third of patients had an objective response, and those are fancy words for saying what percent of patients show scans that have a certain amount of improvement, and there are a set of rules that we use to measure and compare scans to determine these measurements. This is a table listing the side effects, and most of the side effects are in relation to the chemotherapy. So there are certainly some from the interleukin-2, but essentially none or very few that relate to the cells themselves. And you might expect that because these were cells that were in the patient's body before the processing. Again, they're in large numbers, and so maybe that might cause some differences. But by and large, the side effects of this treatment come from the conditioning regimen chemotherapy and also the interleukin-2. The package insert, so that's the FDA description of the trial, includes some stern warnings that some patients can die as a consequence of the chemotherapy. That is true, but again, in centers that have experience giving this form of therapy, it can be offered relatively safely, but there's no free lunch in this or any other aspect of oncology. So who should get lifileucel adoptive cell therapy or TIL therapy? Patients with unresectable or metastatic melanoma who've had growth of their melanoma despite an immunotherapy containing a PD-1 medication and/or targeted therapy for those patients whose melanoma has a BRAF mutation. There are other qualifications or requirements. So you have to have a certain amount of melanoma that the surgeon can remove to process the immune cells. You have to have relatively good functional status, so-called ECOG status or Eastern Cooperative Oncology Group status. And so sometimes I refer to this in the clinic as the eyeball test, or basically how robust patients appear when they come into the clinic. This is not a treatment for patients with active infections. And also, patients have to have relatively normal cardiovascular status to tolerate the interleukin-2 part of the therapy. So, as Chris mentioned, most of my career has been dedicated to clinical trials work. And so with almost every single talk I give, I make some mention of new ideas that we and others are working on. And so in the surgical specimen for tumor harvest, there are quite a few different varieties of immune cells there. And so Dr. Andy Weinberg, who works in our institute, his lab determined that a particular subset that may only comprise a few hundred or 1,000 or two cells in a tumor specimen have the very greatest abilities to fight the cancer. And this is true not just in melanoma, but in other tumors. But we think that there are ways, again, to select TIL that might have greater anti-tumor abilities. You can add genes into cells now, which is awfully cool, but you can give them new properties that they didn't previously have. And this has been used in a variety of tumors. Dr. Ram Leidner and Dr. Eric Tran in our group have been working on this in pancreatic cancer as well as other cancers, but this technology could also be employed in melanoma. And there are also ways to alter T cells to make them hardier when they encounter the tumor in microenvironment, which is a very hostile place, which has very little normal nutrient balance. It can be very acidic. It can have low oxygen. And so those things are advantageous to the tumor, but not so much to the immune cells. And so their technology is being worked on to make the immune cells, again, able to function and to get rid of cancer. So here are some conclusions that I would offer: that TIL therapy works after a lot of work that spans over 4 decades. Lifileucel TIL is the only FDA-approved therapy for selected patients with advanced melanoma after they've had progression on checkpoint immunotherapy or BRAF-targeted therapy. And there's still plenty of work ahead and room for improvement with this technology. Thank you.